Let's start the program with lamp lighting ceremony. I request Lakshmi Manugna for a prayer song.
या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रान्विता या वीणा वरदण्डमण्डितकरा या श्वेत पद्मासना या ब्रह्माच्युत शङ्करप्रभृतिभिः देवैः सदा पूजिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापः.
Thank you, Manugna. Our principal, Dr. A. Ramesh, has been associated with VIPER since 2011 and has made significant contributions to the institute.
Good morning. So, as you all know, you don't require my introduction, right? Of course, people from live, they require. But anyway, to cut short and make it simple. So, today we are very happy to have with us the esteemed and elite professor, Dr. Nagano, who is one of the very good teacher and a scientist with lots of patience. So I think we as teachers also should learn from him. I told him very clearly, you being such a big person, he was sitting from morning 9 o'clock till evening 6 o'clock every day, with accompanied by Mrs. Eringa, right? So, which we teachers doesn't sit and guide the students so much. So I was telling you, we should learn a lot from you, and you have a lot of patience, right? So thank you so much, Dr. Nagano, for blessing our students. And so I think you can see his gesture, right, Mano? Okay. So anyway, I think there are many lessons which we have learned from him during his stay here. And when Santosha has gone to Japan for training under their university under him, right? She was telling me how it has transplanted and the variation between our system and their system. And you'll be surprised since our students, of course, our SVCP, Bhimvaram faculty, madam has gone there. And morning 6:45, they used to come to the hostel to pick them up to the university, which is just walkable, right? But they used to come personally, take them, and again leave them in the evening 7 o'clock. So that is how I think we all tell you the difference between a 5-star and a 3-star hospitality. So I think they have got 7-star hospitality, I believe. So I think we should learn many lessons from them. Although uh, that made me attract a lot. And yesterday I was with him throughout the day, touring him around Hyderabad, and it gave him a very exhaustive trip, I believe. So he was telling too much tired, right? So I think let us learn from him, right? And although I think this topic is something not related to us, right, or which is new to us, but let me tell you one thing, right? This is something beyond our curriculum, and this is how we should stand out from the rest of the crowd. And secondly, we have Mrs. Eringa, who is right a student from Japan working in sorry, student. I mean, her homeland is Sri Lanka. Now working in Japan, getting settled there as a scholar, and she has been here for almost 14 days till now, and she is leaving tomorrow. So I think you should have seen her every day, right? And I think we all should learn from them the way we are dedicated to the work discipline. So thank you, Eringa, for staying here. I hope you got each and every inch known in this campus by now, right? So she's also speaking about Pushpa too, and she has got all that knowledge also. Yesterday, I was surprised to know that she's a fan of Ram Charan. Ah. I know. Okay, right. So jokes apart. So thank you for being with us and making this very fruitful. And last but not the least, the important person, the backbone in making all this possible is Dr. Raju Edala, who is Dean for R&D for the entire society. So he is instrumental in bridging the gap between the Japanese government and our government and ensuring to lure the funds for the transfer of our students from here to there. So that way, I believe that thank you so much, Dr. Raju, for always being with us. And have you all seen him anytime? Yes, no. So I think you should be regular here, right? Okay, good. So I think he is instrumental in now sending 7 more students in January from our engineering stream. So I think we are very happy to ensure that he has plans to send our students also, right? But it all depends upon the project, the passionate people, finding the right people, and so on. So I believe we have already entered into the system of getting transformed on either side. And in future, as per my knowledge, they have applied for a couple of funds, which they will be only sharing you. So I think the reason why we ensure that we have taken their precious time out of the workshop going on for 15 days is to make you understand that there are opportunities globally, not only in US or UK, right? But I think uh, they will share you more regarding this. So with this, few words, thank you once again for all the team for spending your valuable time, and I wish you will have a very good memories to take back to Japan tomorrow. Okay, so once again, thank you all, and advance New Year wishes to all of you. Thank you.
It is with great pride I introduce Dr. Raju Edala, a renowned scientist, environmentalist, academician whose remarkable contributions have significantly advanced the fields of remote sensing, digital image processing, and geographic information systems (GIS). Dr. Edala earned his PhD from National Institute of Technology. He served Dean of Research and Development at the Vishnu Educational Development and Innovative Center, Vedic, in Hyderabad, India. Among his many contributions, he been honored with the Scientist Award and the Green Environment Award, and his innovative work and dedication to the environmental conservation. He is also the founder of Cafet Innova Technical Society, which promotes technical education and research. Please join us in welcome the visionary award-winning scientist, Dr. Raju Edala.
Respected Principal Professor Ramesh Garu, Dr. Yukio Nagano, Associate Professor Saga University, Japan, Mrs. Eranga Pavani Vitarana, Researcher from Saga University, Japan, faculty members and dear students, very good morning to one and all. Very happy to share this podium with you all along with the Professor Ramesh Garu and the guests from Saga University. This is an important day for you all because you are going to interact with the great scientist and an academician from Japan. If you see him, the innocence, the hard work he used to carry in his career is really unremarkable. So he's a great scientist, is very friendly to interact with the students and the faculty members. Has been more than 8 years of close association with Professor Yukio Nagano. Through my professor from Kumamoto University, Japan, is Professor Takashi Watanabe. Last week he was here with us for the international conference in BBRT Nasapur campus. So it's a great team, great scientific team. They are here and they're coming all the way from the Japan to interact with you and collaborate with you all. It is a great opportunity the Sri Vishnu Educational Society is providing under the great visionary leader, our beloved chairman, Sri K.V. Vishnu Raju Garu and Mr. K. Ravichandran Rajagopal Garu and our Aditya Garu, secretary and vice chairman. So you are all lucky to build your career in right ways. Yeah, I do agree with the principal. He has almost already covered all the topics, why we are here, why they are here with us, and what way we are going to collaborate with the Japan. Yeah, we are very lucky, and uh, more than 6 months, we are closely associated with the research in terms of submitting the collaborative research proposals and also visiting our faculty members to the Saga University and stayed 12 days under the Sakura Science Exchange program, fully sponsored by government of Japan with the organization called Japan Science and Technology Agency. Like what we have DST, Department of Science and Technology, they have Japan Science and Technology Agency. It is fully sponsored by the JSTA. Through the professor only it is possible. Now in the next month we are going to 8 members. We are going to visit Kumamoto University. That way we are bringing a lot of opportunities for you to build your career and settle in a good way in the abroad for higher educations and the job purposes. Please come forward with your capabilities and try to interact with the scientists. And try to collaborate with them, express your interest and express your knowledge and everything. Try to collaborate. Then we are here to support you for your better career. And you see here the young researcher from Mrs. Eranga Pavani Vitarana. She's basically from Sri Lanka. Now she became a very good researcher after joining Professor UK Nagano's laboratory. She mentored our faculty members and students, even in Japan, and now also from 18th onwards, she is staying with us. And very thankful to Dr. Santosha Madam. Your morning to evening, you're accompanying her. It's not that easy. You both are staying away from the family and all these days staying in our Narsapur guest house and supporting her and taking care of her is not that easy. Thank you so much for your great hospitality and support, ma'am. And last but not least, I would like to thank again Professor Ramesh Garu, Principal, Dr. Santosha Madam, and the faculty members of Pharmacognosy, the head of the department, Professor Lakshmi Madam, and other faculty members. Thank you so much for your great hospitality. And you cannot believe, I am seeing the principal very friendly and great principal, I can say. It's not that easy, you know. I will tell you one thing. Yesterday was Sunday, right? He came all the way and he spent complete day with the two guests. From morning 10 o'clock to night 10:30 he was with the guest. Nowadays, who is the principal will stay like that. See how much importance he is giving for the guests, and he is trying to bring those resources for your his faculty members and students. You are really lucky to have him as a great principal for VIPER. This, thank you and have a great interaction with both of the researchers. Okay, try to find your good outcomes for your career development. Thank you. Thank you. Have a great day.
Thank you, sir. It's my great pleasure to introduce our speaker for this session, Ms. Eranga Pavani Vitarana. She is an accomplished researcher in the fields of genomics, bioinformatics, and molecular biology, currently pursuing a master's degree in Saga University, Japan, under the prestigious MEXT scholarship awarded by the Japanese government. I request ma'am to address the gathering.
Good morning, all. Mm, It's nice to meet you all. This is my first experience in India. Mm, I am originally from Sri Lanka, but this is my first experience in India. I want to really grateful to the your madam Santosha. She is always with me and supporting me. Earlier, I was worried about this visiting India because I'm scared to. I came here alone without my sensei. So, but uh, she supported me a lot. I feel a lot of comfortable, and I want to thank your principal sir and Dr. Raju. This is a great experience to me, and also it is a remarkable event in my life also. So thank you so much.
Thank you, ma'am. Now I request Dr. Yukio Nagano sir to address the gathering. Yukio Nagano sir, working as an associate professor for the Analytical Research Center for Experimental Sciences, Saga University, Japan. He holds a Doctor of Agriculture degree from Kyoto University. Dr. Nagano research expertise spans biodiversity and systematics, genomics, applied molecular and cellular biology, and applied biochemistry. I request sir to speak few words.
I'm happy to coming here, and thank you so much for inviting us. And the primary purpose to visit here to perform workshop for genomic research, and some of them, these tools are now joining. But I'm very happy to have an opportunity to do this one day international symposium, and maybe many the undergraduate students maybe. So I will talk about some basics of our previous research today. And in addition, maybe this one is more important one, is that I will introduce how to study in Japan. Okay. So please, and maybe one problem in this university is you do not perform questions. So please raise your hand and give me your questions to me. Okay, this is very important because I do not understand. Uh, I do not understand your situation, so please raise your hand and give me questions. Okay. So soon we will perform presentations, so wait for a while. Thank you so much.
Thank you, sir. It gives me great pleasure to welcome you all to this session. Today we have gathered here for a session on frontiers in plant genomics, DNA sequence analysis, and student career pathways in Japan. Such sessions provide an excellent opportunity to collaborate, connect, and grow together. Moving on to the session, Dr. Yukio Nagano, working as an associate professor for the Analytical Research Center for Experimental Sciences, Saga University. With a distinguished career, Dr. Nagano has held academic and research positions at Saga University, Nagayo University, and Shiga University in medicinal sciences, contributing significantly to life sciences research since 1992. He has authored 83 research publications and has successfully led 17 research projects funded by the Japan Society for the Promotion of Science, JSPS. Dr. Nagano also serves as an editorial board member for scientific reports, highlighting his international recognition and contributions to the scientific community. His work integrates molecular biology and applied biochemistry. Please join us in welcoming the visionary Dr. Nagano onto the stage.
Are you okay? Okay. So today, how to do it? Oh, today I will talk what can we learn from genome sequences and what discoveries. Oh, and sorry, what discoveries have we made from genome sequence? Okay, and here you can watch many faces, but you understand that these are humans. Okay, what you do, you find that many faces are different. Okay, and so small differences in DNA sequence account for differences in appearance between one individual and the next. Okay? Therefore, to study these to study these phenomena, study of genome is important. And maybe many the undergraduate, so I will introduce, maybe this one is very basic one, but I will introduce what are DNA, genes, and genomes very shortly. Okay, so these are cells, many types of cells. and these are brain cells, and the protos are green. And inside the cell, there are DNA, genes, and genomes. And maybe you know these terms, but you do not distinguish these three terms. So these three terms are important: DNA, gene, genome. And they are similar words, but their meanings are different. Okay? DNA is a sequence of molecules. So maybe you know this type of figure. Maybe I used the three ways to show DNA sequence, and maybe you know that DNA is double helix. DNA is double helix, and GC and TA pairings are present. So DNA is a sequence of molecules, but it's not real. Real letters are not present, but these molecules are shown by GCAT. And so DNA is composed of 4 letters, GATC. So DNA is similar to this. This one is alphabet. And this one is music code. And this one is Morse signal. And this one is Chinese letters. And this one is DNA sequence. Okay, so DNA is a sequence of molecules. And gene is a meaningful sequence. So DNA and gene is, so gene is similar to sentence. Okay. So here, these are DNA molecules and genes are located here. And by the transcription and the translation, protein is created. So in other words, a meaningful sequence means a sentence in terms of holding information to make proteins. So information of protein is included in genes. And these proteins play very important roles inside the cell. So protein has too many functions, and they play very important roles. So difference in gene result in difference in protein function, and difference in protein function results in difference in trait, such as various properties of organisms. So maybe many these individuals are different, and these are caused by differences in genes. Okay, so you are different face and different properties, but these are caused by differences in genes. And differences in traits produce difference between individuals and difference between organisms. So in the case of human, differences are present, but their differences are small. But when compare human and monkeys are here, but there are many differences in DNA sequences between human and monkey. Okay, so these are produced by the difference in DNA sequence and difference in genes. And the final one is genome. Genome is similar to a book or a blueprint. Yes, so by combining genes, chromosomes are produced, and all chromosome is genome. Okay, and DNA, gene, and genome has two aspects: books, sequence and letters are both materials and information. And similarly, genomes, genes, and DNA are materials and information. So, okay. So finally, difference in genomes produce difference in trait, and these differences produce differences between individuals and differences between organisms. Okay, so these are produced by the differences between genomes. So now, by using high throughput DNA sequencer, genome analysis become affordable ones. So now we introduce our research group discovery, but our main topics is Eranga's one, and she will speak our very recent topics by herself. So we are analyzing many organisms, fruits, and the other plants and fungi and insects. But now, today, we want to focus on citrus and its relatives. Maybe these are so many maybe many students are from VIPER. So now today I'll introduce relations to health here. This one is so chimpi in Japanese and chempi in Chinese, but letter is identical, is used as traditional medicine in Australia. And but this one is Hong Kong, but we can get it in market. Okay, in Japan, sun-dried peel of Satsuma mandarin, citrus unshu, is known as chimpi. Do you know this one? Yeah, do you know this one? Here? This one is available here. Orange peel, available here. Available here. Oh, okay. And one way is to use ingredient of this spice. This one is called Shikimi Togarashi, and this one is traditional spice mix. And chimpi is a common ingredient in Shikimi Togarashi. But the main ingredients are chili, but chimpi is included. And hesperidin is an effective component of chimpi. So, and chimpi has many effects, blood pressure lowering effect, and aromatic stomachic, and many others. And mandarin orange, this one is called mandarin orange, and mandarin orange citrus reticulata is used for making chimpi in China. But in Japan, this one and this one is a slightly different one. And Satsuma mandarin, citrus unshu, is used for making chimpi in Japan. I want to talk about the origin of citrus unshu. So after the appearance of chimpi, this Satsuma mandarin appeared. And this one is original tree. Original tree is still present. And this one is located, this plant is located in our university, and it was propagated through mainly by grafting from this tree. So this one is written in Japanese, but over about 300 years, various cultivars, so cultivated varieties, have been developed from original tree, and many cultivars are present. But they are produced from asexual production. So without performing sexual production, these many cultivars are produced. So some branch is changed, and that branch is used for as a next one. And by using such method, there are too many cultivars are present now. So despite its relatively short history, the creation of various cultivars is what makes the Satsuma mandarin and other citrus fruits so appealing. But my evaluation of gene is that I must say the effects are probably limited. However, citrus fruit itself have a deep connection with human health. One example is scurvy. This type of, I do not know this disease details, but this was a famous disease. And during the age of exploration, so their movement of Western people and scurvy frequently appeared. But this problem was solved by citrus fruits. And later, not this time, but later, it was discovered that the cause was a deficiency of vitamin C. So, citrus has vitamin C, and storage of orange is very easy. So you can keep citrus for a long time, such as sweet orange, lemon, and others. One mystery is here. This is the history of this ethnic group, Austronesian language family, which are from Taiwan, and they moved to many places from here, but not to Japan, but they moved to many places. For example, Madagascar is occupied by these peoples. But similar problem, they may have similar problem, but this problem has not been solved. But they may bring stress for their travels. So this was one of mystery. And this was a bad example. Stress is used for pseudo medicine. This is bad example. He is Linus Pauling, and he studied chemical bonds and got Nobel Prize in chemistry, and he performed peace movement and got Nobel Peace Prize, and he had many important contributions. One is protein structure, alpha helix, and he found molecular evolution. And molecular evolution is the basis for genomics. So his research work is too great. But she also established also molecular medicine. And he said vitamin C as a magic drug. And usage of vitamin C for medicine causes many problems. So this is a bad example. And another bad problem is grapefruit and pomelo. Grapefruit is a hybrid between the sweet orange and the pomelo. Okay. And this plant produces many amounts of flanocumarins. And flanocumarins are strong inhibitor of P450. So P450, cytochrome P450 is very important for drug metabolism. And this compound inhibits P450. So Furanokumari has strong interactions with drugs. So this product produces some problem. And another important one, I think, the usage of stress is, I think this one is important. Shimitoragashi is important. And its ingredient is peel of orange. So next I want to speak about, and I will talk this story later. And now I want to talk the history of citrus. So this region, Assam is here, and Kachin and Shan is here, and Yunnan is here, and these places are considered the origin of citrus. But here, from Philippines to Japan, this side, there are Tachibana. The other type of citrus are present. One is Tachibana, Japan mainland, and this one has two types, Japan mainland type and Yukio type, and Korean Tachibana. And this one is located in Jeju, this island, and here, Yamaguchi prefectures, and Shikuasha. And these are also related to the origin of current citrus cultivars. But basically, four basic species, mandarin, papeda, citron, and pomelo are present, and plus others are present. Plus others are kumquat, ichan papeda, and some indigenous citrus from various regions, including Japan. But these four are mainly important, and by the contribution by these four species, many types of citrus were produced. For example, Mexican lime is produced by the hybrid between citrons and micranza. Micranza is papeda, and by the hybrid between sour orange is produced by the hybrid between mandarin and pomelo, and by the hybrid by sour orange and citron, lemon is produced. And Lampure lime and rough lemon is produced by hybrid between mandarin and citron. So please remember these origins, because I will talk about soon about this. And one famous one, sweet orange is very complex, but they are produced by the hybrid of some type of mandarin and pomelo. Okay? So Lampure lime is produced by citron and mandarin, and rough lemon is also produced by citron and mandarin. And our genome analysis found that Lampure lime, which is not supposed to be in Japan, is growing in Yukio Island. And but us we believe that this one is sequencer that is native in Japan. But by performing genome analysis, we found this plant is Lampure lime. And these holes are important. But we found that citrus Nippo means Japan, and Korean means Korea, and this one is called Korean Tachibana, and this plant is native to Japan and Korea. But gene flow from Japanese cultivated variety produce Kimikan or Ogonkan, and now known as Citrus flaviculapas. But these are produced from native species in Japan. So this one is one of our discovery based on genomics, but not published. And we also find another hybrid between mandarin and citron. So Rampure lime and rough lemon is produced by the hybrid between mandarin and citron, but we found another one. So he is Cherin Penjol from Bhutan. Bhutan people is similar to East Asians, so it may be you consider these two the Japanese, but he is from Bhutan. And our discovery was introduced by TV program in Bhutan, and our discovery was appeared in the front page of one of the scientific journal. And here is Bhutan, and maybe you know here is India, and this is a beautiful place, and there are many rivers, and now to produce water power, many Indian workers visit Bhutan, and now they are producing dam to produce water power. And here is a beautiful temple. And Mexican lime is well-known lime, and this one is produced by hybrid between citron and papera. But we found new type of lime, and we named it as Himalayan lime. And Himalayan lime is hybrid by citron and mandarin. And this is our discovery based on genomic science. Okay, another story is Xanthosardium. Xanthosardium is a member of citrus family, but not members of citrus subfamily. Okay, and maybe this one is very famous in this country, but I do not know how to call it in Telugu. But this plant is used for Ayurvedic medicine. Maybe you are familiar with this problem. And they have many effects, and such as stomach pain and snake bite and others. And another famous Xanthosardium species is called Huajiao, and this is a key component of Sichuan cuisine. So they use it for many purposes. And this one is called Mapo tofu, and this one is very famous dish. And Chinese people use similar purposes for as medicinal use. And Sansho, Xanthosardium pipericum, is grown in Japan, but except Yukio and Korea. So these are called Japanese pepper or Korean pepper. And in Japan, production is decreasing due to the aging of farmers. Therefore, recently, major Japanese food companies have added Sichuan pepper to their sansho product. So this is a problem for me. But he is a Chinese man, and he visited about 1 year ago to our university, and now he is attempting large-scale cultivations in China. So I'm looking forward to the result. And sansho is used for so many purposes. One is this one is sansho, and this one is eel food. And by the combination of sansho and this one is important in Japan. And in this case, we use flowers to make hot pot. And we use this one for many purposes. And as medicinal purpose, we use usage is very similar to China and India. And one product, kumichinki, is kumichinki include sansho, Xanthosylum species. And I think this is important feature. So sansho has a salt reducing effect. Okay, so rather than medicinal use, this effect may be more important. So we analyzed many, more than 50 sanshos, and we collected many samples from these regions and other regions, and found that many are, and we discovered many, but it's very complicated. So I want to explain most important points only. And he's Dr. Tomitaro Makino, and he's called Father of Japanese Botany. Okay? And in his literature, this is his previous paper, and orientation of letters is this orientation. And this is a mixed language of Japanese and classical Chinese. So, but to enter university, we must study classical Chinese. So I can read these very old letters. And he named the spineless sansho as a subspecies of Xanthosylum piperitum, and named the Asakura sansho, and he named some subspecies name. But we found that there are many types of spineless sansho. So spineless sansho is not a monophyletic lineage. So therefore, spineless sansho is not a subspecies. And another finding is we found that domestication is underway. As a first stage, from mountain to field were observed. These are three examples. One is Asakura sansho, and two lineage of Arima sansho. In addition, we find that the spread of superior lineage. So one of Asakura sansho, but this one may be Korean origin, is spread to various places, and Budo sansho is also spread to several places. So two steps from mountain to field, and the spread of superior lineage were observed by the genomics science. And the next question, and this is a final topic. Is it possible to connect research on citrus and its relative to the improvement of Japanese cuisine? So now I want to focus on problem in Japan, but this story may be linked to Indian problem. So now here's the global situation. Maintaining nutritional balance while restricting calorie is most important for our health. So eating many has a problem, but balance is important. And now excessive calorie intake is a global issue. And maybe this problem has been already happened here. And the success of Green Revolution, and now we can get rice wheat very easily, and price is very low. Success of Green Revolution and the spread of conventional agriculture, not organic agriculture, have contributed to this. However, hunger problem remain, especially in sub-Saharan Africa. And this one is calorie per capita per day. And this is Japanese level. So in Japan, maybe level is similar in Japan and India, but Japanese people may be more smart, I think. That is question. So I think calorie may be lower than India because many Japanese are smart. But in Japan, excessive calorie intake has not become a major issue. So many developed countries and China, now excessive calorie is problem, and this number is world average, but we eat less amount of calories, so color is similar to these countries. So, but balance of nutrients is maintained. So in Japan, this program does not become major issue. And now the magic drug is blockbuster is appeared, and blockbuster now obesity drug is produced, and this one is very effective. And now the companies in India and China are making lower cost version of this magic drug. So by the appearance of new drug, global issue of excessive calorie intake might be resolved, probably. So now our Japanese cuisine have some problem. One is excessive salt intake. So one reason is we use fermented food. And fermented foods are claimed to be healthy, but they also contribute to excessive salt intake problem. So this is one problem. And we use soy sauce and miso, and these contain many salts. And calcium deficiency and iron deficiency is another problem. Because our Japanese do not eat, Japanese can eat many type of meat, but we do not eat much amount. So maybe calcium deficiency and iron deficiency is now problem in Japan. My question is how to address the issue of excessive salt intake. So we should encourage the active use of spice. Another important one is called umami, such as umami, sodium, なんだっけ、sodium glutamate is important for sodium glutamate also has salt reducing effect, but spice has salt reducing effect. So this spice called shimi togarashi is one important one. And xanthoxylum is important one. And sansho's salt reducing effect is also important. So I think this is important. So we should promote the Indianization of food in Japan. So we want to study some spices and how to use spices in this country. But problem is excessive consumption also have some problem. So this is a problem. But I want to study how to make dish here. And I think this Indianization of food is important, but the problem is that many Japanese cannot eat spicy foods. This is a problem. Okay, thank you so much.
Does anyone have any queries? You can ask.
Raise your hand.
Good afternoon, sir. This is Shyam. I'm a final year student. Yeah. Previously, you have mentioned about Austronesian language family.
Hi, hi.
Yeah. What does it mean?
ね、So in this figure, you understand some Western people move all over the world by ship, and similar problem may be present. Another problem is here. So from Taiwan, many people move to many countries by using ship. But there is no record, but based on similarity of morphology and similarity of DNA, first some Taiwanese people moved to here, and then moved to many places by using ship. So similar problem, deficiency of vitamin C should be present. But we do not know how they solve this problem. So this is unresolved question. So I want to solve this problem. So they may use citrus to get vitamin C. So this was one mistake. So I want to solve this problem. Okay.
How does Shansho has what's the mechanism of Shansho in salt reducing property?
I don't know because mechanism is not solved, but generally using spice and using umami. Umami is called, one famous umami is sodium glutamate, has salt reducing effect. But mechanism is not solved. But by adding small amount of sansho, so by reducing salt and after adding sansho, salt level, so by our tongue, salt effect become higher. So such phenomena is observed. So by reducing salt amount, but by adding sansho, and we feel that this is salt.
That property.
So as a phenomena, salt reducing effect is observed, but we do not know we do not know mechanism. But maybe the taste receptor may be involved in this phenomenon. Okay.
Thank you, sir.
Thank you, sir, for your valuable session.
But I will talk about study in Japan later. But before then, she will perform presentations.
It is my great pleasure to introduce our next speaker for this session, Miss Eranga Pavani Vitarana. She completed her Bachelor of Science in Agriculture Technology and Management at the University of Peradeniya, Sri Lanka, where she laid a strong foundation in life sciences. Her research focuses on phylogenomic insights using whole chloroplast genomes and nuclear genes, where she has developed a cost effective workflow to explore phylogenetic disorders. This innovative work reflects her passion for applying advanced molecular techniques to cutting edge scientific problems. Her academic journey includes collaborative research experiences and the guidance of esteemed mentors such as Nagano sensei at Saga University. Soon she will be graduating and holding her doctoral studies at the United Graduate Schools of Agriculture Sciences, Kagashimo University. I request ma'am to take over this dais.
Good morning, all. Uh, First, I would like to thank all the organizers of this one-day symposium. I'm a little bit sick because of cough. Don't worry about that. I will try to do my presentation in my best. Okay, today I would like to share my academic journey and my research in Japan. First, I would like to briefly explain about myself. I am Meranga Pavani Vitarana from Saga University in Japan, but basically I am from Sri Lanka, where I completed my bachelor's degree at University of Peradeniya. Now I would like to share with you my journey. Actually, during my undergraduate life, as we all faced the COVID-19, so at that time, I was the last year student at University of Peradeniya. So at that time, I was doing my final year project, but because of COVID-19, I couldn't able to go to lab, so all study was halted. So therefore, I had to stay at home, and I had to find a study in order to graduate. But at that time, it was challengeable to me and also my local supervisor, how we completed this study and how we need to graduate at time. So in that period, we faced lots of difficulties, but as every difficult situation create opportunities, I got connected with Nagano sensei, and I learned about bioinformatics. Actually, at that time, my local supervisor also didn't know about this type of studies. So it was huge challengeable to me. There's no one to guide me. So I learned all these things by myself, by reading textbooks. And I am very lucky to have Nagano sensei because he always motivated me and guided me. He provided the textbooks. After opening those barriers, he sent two textbooks to me to read before starting learning this bioinformatics. So that's why I'm telling I'm really lucky to have him. And I think you also really lucky because you now you all also got connected with Nagano sensei. So don't miss this opportunity. That time I completed my bachelor's study and I was graduated. Then I started collaborative study with Nagano sensei. As the first step, we started this chloroplast analysis for citrus and its relatives. So this is the turning point of my life. Then while doing our collaborative study, he invited me to apply for the scholarship in Japan. So there are lots of scholarship in Japan. So one of most important one is Japanese government makes scholarship. So we applied for this MEX scholarship, and then I got awarded from MEX scholar. This MEX scholarship covered all the tuition fees and all the living expenses in Japan. So it is the great opportunity to me, awarded as a MEX scholar, because it covered our all flight fare tickets, all things. So I think Nagano sensei will later discuss about these opportunities. So I am not going to explain this opportunity, but in my experience, I want to highlight MEX scholarship is the best scholarship in Japan to study. I came to Japan in 2022 in October. As a research student, I firstly enrolled in Saga University. You can see some picture. It's not working. Okay. Not only doing research things, we can enjoy our life in Japan. This is the first year I visited the Saga International Balloon Festival, and I want to mention I have a best supportive lab friend. She is also from foreign country. She's from Myanmar. She also very supportive lab friend. So I am really lucky to have her with Nagano sensei. So we visited this temple in Fukuoka. Your ma'am also visited this temple in last year. So okay. Then I enrolled as a master's student from 2023 April. You can see lots of pictures. I want to motivate to you to get this type of opportunity and learn in study in Japan, because as I experienced, Japan is the safest country to go. And actually when we are doing our studies, sometimes it will finish around late night, so we can go from after finishing our works, we can go to our apartment, to our residence without any fearing around early morning 2 or 3 like that, because Japan is the safest country. And also we can join lots of conference, and we can make international friends. And I think you can see this is the Sakura Science program. I and my Japanese friends assistant, they are training program in Japan. So we can engage lot of activities. We can taste Japanese foods, but still I am not good at tasting Japanese food because of some sauce taste is not good for me. And some are worrying about before going to Japan, we need to learn about Japanese, but it's not essential. In my case, I didn't know any Japanese, actually in any Japanese letter before going to Japan, but I know now little. We can learn from their environment. I'm not good at Japanese very well, but I know little, I can handle. So with that environment, we can learn Japanese. We don't need to any Japanese skills to get the next scholarships. So don't worry about the language barriers. Now Japanese, most of the Japanese people can speak English. They also want to learn the English language. So don't worry about these language barriers. We can handle it. So, so I want to mention you, from my experience, I am also not this type of person in the beginning, but I think my skills and my knowledge, all things were polished because of the Nagano sensei. So I want to tell you, take this type of opportunities, believe yourself, and uh, believe yourself, get that type of challenges, believe yourself, work harder, you can make your dreams reality. As the next step, I would like to share with you my study. Uh, This is the recent publication, subfamily evolution analysis using nuclear and chloroplast data from same reads. This study is actually accepted by Nature Scientific Reports in recently. We hope it will available in online very soon. But we already published our preprint, so you can access using this QR code for our to read our full publication. So first, I would like to explain why should we study the evolutionary relationship between species. Evolution is not just a concept; it is the studying of the story of life on Earth. We can know the relationship between species, how they are related, how they diverge over the millions years, and also we can predict the future changes. For example, we can study the how organism adapt to the environmental changes in the past, and we can predict how they might current or future environmental challenges. And also it has real world application in science and society, including agriculture and pharmaceutical industry. We can use this knowledge in order to identify, uh, in order to develop new drugs. We can find bioactive compounds in relative species, and we can find novel compounds in the relative species, and we can find alternative sources. So studying the evolutionary relationship between species is very important. Uh, for the analysis of phylogenetic relationship, we have several type of method. One method, one trendy method is use of chloroplast genome sequences in plant phylogenetics. Why chloroplast genome? Because this chloroplast genome has typical maternal inheritance, conserved gene content, low recombination rates, and manageable size. Because of these characteristics, make them excellent choices of phylogenetic analysis. But there are and overcome these issues, scientists focus to extract large number of nuclear genes integrate in order to tackle the develop a cost-effective workflow by using same row sequencing reads for both chloroplast analysis and nuclear gene analysis. Here, for the nuclear gene analysis, we have used read-to-tree method. This read-to-tree can extract conserved nuclear gene sequences from the raw sequencing reads directly. So this is the advantage of this read-to-tree tool. Compared to other tools, most of the other tools are depend on targeted species and customized databases. So in this case, read-to-tree is a reference guide approach. So it is the unique advantage of the read-to-tree method. In read-to-tree, we can identify two types of output. One is concatenated nuclear gene tree. Although the validity in plant phylogenetic is not still tested, and the second one is multiple alignment of each as an ideal candidate. We choose subfamily Aurantidae. Now you might be wondering why we did select this subfamily Aurantidae. Let's see. I think you know these species: lemon, lime, mandarin, pomelo, citron, orange, and grapefruit. These species are citrus species. Other than these citrus species, orange jasmine, curry tree, tanaka, bale, and wood apple, these are the citrus relatives. So economically and nutritionally important species. I hope you all know this disease. Disease is scurvy disease. This cause of lack of vitamin C. During the 18th century also, people used these limes, lemons, and orange in order to treat this disease. Other than because these species contain high rich of these species are rich of vitamin C. Other than this, these species can be used in culinary, household cleaners, cosmetic, and personal care, agriculture uses, digestive health, and cancer prevention. So there are lots of benefits in these citrus and relatives. The second reason is our previous study also focused on this subfamily Aurantidae. So by using nuclear DNA sequences. Therefore, by revisiting this subfamily Aurantidae, we could able to use this results as a reference in order to validate our read-to-tree results. So as the first step, we validate our read-to-tree in plant phylogenetics. Here, as we see here, on the left side, you see the read-to-tree result. We see right side our previous result. So interestingly, these two results are aligned well. So this validates the read-to-tree in plant phylogenetics. Then we focus the conflict analysis. Then here we use 39 species. Here you can see the nuclear gene tree and chloroplast gene tree. As we see, there are conflicting signals between these two trees. So we can see inconsistencies within the genus and between genera. For example, here you can see this group, Citropis and Naringi, is closely related to the Atalanthia group and Citrus group. But based on the chloroplast results, you can see this Citropis and Naringi group is closely related to the Citrus, Atalanthia, and Feroniella and Limonia group. So we observe this type of conflict signals between these two analyses. But we observed that consistent patterns of subgrouping. So this highlight the need of refined classification for this subfamily. Therefore, we propose novel classification for this subfamily. So we divided this subfamily into two primary groups as group A and group B. In previous classification, this subfamily divided into two subgroups as Citriae and Closinae. But in our novel classification, we moved this Muraya group, which is categorized under the Closinae subgroup in previously. We moved this Muraya group into group A. So now group A includes 6 subgroups. True citrus fruit trees. Here we categorized all the citrus species as true citrus fruit trees, and all the Atalanthia groups we name as Yakinaram, because in this group includes the Atalanthia selanica, which is commonly used in Sri Lankan Ayurvedic medicine. Then we group this Citropis and Naringi as Tanaka because of the Naringi crenulata. This Naringi crenulata is commonly called as Tanaka tree, which is popular in Myanmar as a beauty aid. Then the Feroniella and Limonia group we name as wood apple, and this Paniculata and Marmelos we name as bale fruit, and all the remaining species we grouped as group B. So next, let's see the read to tree for conflict analysis. Here is the workflow for this conflict analysis. As I previously mentioned, read to tree produce two type of output. One is concatenated nuclear gene tree. Other one is multiple alignments for each gene. So this is the important output from one of the important output from read to tree method. So we use this multiple alignments for each gene. Tree. The read to tree produce more than 16,000 gene alignment. Therefore, in order to identify best gene alignment for the downstream analysis, we have applied a quality criteria. The ratio of ambiguous N characters to total nucleotide had to be less than 10%. By applying this criteria, we selected best 579 gene alignment for the downstream analysis. When we are observing these gene alignments, we observe that all these gene alignments have missing data consistently under 20% for each sample. By using these gene alignments, we generated species tree and gene trees. So now let's see the conflict analysis in species tree versus gene trees. So here you can see the results for this conflict analysis. In this pie chart, the blue color shows the concordant topology. That means similar topology, similar appearance of the gene trees and the species tree. And this red color shows discordant topologies. That means the conflicts between the species tree and gene trees. Using this results, we identified that most of the conflict occurred in true citrus food groups, Yakinaran, Tanaka, wood apple, bear, Muraya, and the group B. So, in order to perform the discordance analysis, we mainly focus three key areas. One is conflict in true citrus food trees and its near subgroups. And second one is conflict in bale fruit subgroups and its associated species. And third one is conflict in group B. In order to perform discordance analysis for the first key area, we use around 800 gene trees, and for the second one we use 1964 gene trees, and for group, in order to analyze the conflicts in group B, we use 1135 gene trees. So these type of conflicting signals occur mostly because of the genetic differentiation process, introgression, and Asian hybridization. So now let me briefly explain about this concept. Here we see A, B and C taxa. The black line species tree showed close relationship between B and C taxa. But in the orange line gene tree showed close relationship between A and B. So this happened because of the gene don't sort into the species during the speciation. So we call this as a incomplete linear sorting. On the other hand, here you see D, E, F taxa. So the pink line gene tree shows the gene flow between D and E. So we call this type of event introgression. Therefore, this this incomplete linear sorting and conflicting signals across the genomes. Finally, we discovered that most of the discordance we observed because of the incomplete linear sorting. Other than this incomplete linear sorting, we observed that introgression event in true citrus fruit trees and Yakinara in Tanaka group and bale fruit group, and also Paramigia and Tripacia. And other than that, we observed the ancient introgression followed by incomplete linear sorting in these certain clades. So, in conclusion, our study demonstrated efficient approach to the phylogenetic research by in addition, our study highlight that importance of submitting data to the public databases, because most of the Chinese studies, they are not they don't know they don't submitted the raw data to the public databases. Because of that, we cannot reanalyze this type of studies, and it avoid the transparency. But we, in this study, we highlight the importance of the submitting data to the public databases. So that's bring me to the end of my presentation. Thank you so much. I would like to invite you to join our team, and you can get a lot of opportunities in Japan as me. So I want to invite you to take those type of opportunities and work hard, you can make your dream reality. Thank you so much.
Thank you, ma'am. Any queries?
Yes, please.
Good afternoon, ma'am.
Good afternoon.
Yeah, what's the importance of these phylogenetic relationship studies? What is the importance of these
Okay.
studies?
As I told, we can identify by studying phylogenetic relationship how species are related each other and how species are diverged over the millions years. By knowing this type of relationship and how they are diverged, we can find some in when considered in the pharmaceutical industry, we can find such bioactive compounds in those type of species and similar biocompound is compounds in those type of species or some novel compound like that. So we can apply not only for pharmaceutical industry, in agricultural field we can use this type of studies to improve new varieties for crop yield improvement, and we can conserve, as you know there are lots of endangered species, so we can conserve those type of species by knowing this type of phylogenetic relationship.
Previously, you have mentioned about whole chloroplast genome sequencing.
Sorry, can you?
Yeah, previously you have mentioned about the whole chloroplast genome sequencing. Chloroplast genome sequencing.
Yes, in our analysis we use this whole chloroplast genome sequences in order to create the phylogenetic trees, and also we use nuclear genes to create the phylogenetic trees. So by without rely on chloroplast genomes, we can use these two genomes, that means chloroplast and nuclear genes, to create phylogenetic tree and to provide better insight, comprehensive insight for the history. So we can use both type of method without rely on one method, because we cannot tell which one is accurate. Or both methods had its own advantages and disadvantages. So in my opinion, without rely on one method, it is better to integrate these two. We can provide a comprehensive insight for evolutionary history.
You have mentioned about Tanaka tree usage in cosmetics.
Yes.
Yeah, in which cosmetic it is used?
In Myanmar, they are like I don't know about how they make those type of cosmetics. I only know they applied in face for the as a beauty aid like that, as a powder or something like that. I don't know about those type of cosmetics.
Thank you.
Any questions?
Good afternoon, ma'am.
Good afternoon. Good afternoon. Yes, please.
How wood apple is related to citrus family?
Hmm?
Wood apple.
Wood apple?
Yeah.
Okay. Here, these Ferronella and Limonia group together, we are called this as a monophyletic. So this we name as a wood apple. So these wood apple are closely related to this Citra, Atalanthia, and Citropis and Narinja, that means Tanaka group. So what is Yes, this is not just family. This is sub family. We are calling this as a our entity a sub family. These all species are belongs to the Rutaceae family. This is a one group, just one group. So tuple is just one group under this subfamily our entity. Any? Thank you so much.
Thank you, ma'am. As sir will speak about the scholarships available in Japan, over to you, sir.
5 నిమిషాలు. 5 నిమిషాలు. ఓపిక పట్టండి. కానీ ఏం కాదు. వాళ్ళు జపాన్ నుంచి రాలేదు.
Be silent, silent, be silent. Okay, and now I will have brief talk very shortly. I want to study in Japan. So now first I will introduce 10 appealing points of Japan. One is rich culture and traditions. For example, this one is called ikebana, and one flower arrangement. And be silent, okay? Okay, now I'm performing lecture, so be silent, silent. And this one is called tea ceremony, called sado or chado. And in addition, calligraphy called shodo, and no theater and kabuki, and many of which are many of which have history spanning hundreds of years and highly esteemed globally. And another one is technology, sophisticated technology and manufacturing, especially in our country. Maybe this one is automobile and this one is succeed. And this car is produced by Suzuki. Maybe you know this company. And Suzuki company has not been as successful in Japan now, but they succeed here. But this company is originated in Japan. Maruti Suzuki is originated in Japan. And beautiful nature, maybe one appealing point. This is sakura tree. And this one is なんだ紅葉だからこれ this one is autumn foliage, but you can watch this type of trees. And maybe this is most important point. Japan is safe country. And especially you might go to US or Europe, but its safety level is very high. And girls can walk in the midnight generally. So safety is most important point, especially for girls. And food culture, she does not like food culture, but food culture is one of characteristics characteristic feature in Japan. And this one is our ranking best dishes of the world. And the first one is Japanese dish. And its name is curry. Curry is also known curry. And this curry was introduced to Japan via UK, not directly from India, but not directly from India, but from UK. This dish was introduced and developed into uniquely Japanese dish now. And in addition, sushi, tempura, ramen, and wagashi is also important food. And public transportation system is well established. So you can use the super express and highway system. And maybe you know this, anime and manga is important culture. And we have typhoon. Typhoon is similar to cyclone. Typhoon and earthquake has one problem in Japan, but we have building standards and disaster drills and early warning system. So earthquake and typhoon attack Japan frequently, but generally, sometimes earthquake produces this problem, but generally, our country is very safe. And there are many world heritage sites, and this one is famous Buddha in Nara, and some temples are important. And spirit of hospitality is important culture in Japan. And I am this journal, and her paper will be appeared today or soon. And this journal, scientific reports, are largest open access journal. And please focus on here. So these are ranking of the leaders of this journal. So US is the top, but maybe these 5 countries are read this paper. So Japan is one of the top countries in science. But I think now as an editorial board member of this largest open access journal, I feel that India has more higher level than Japan in science. That this is a problem. And now I want to introduce how to study in Japan. So please remember it. Study in Japan. So please type study in Japan, and you can find this page. And you can check language to your favorite one. So type by using Google Chrome, type study in Japan, and you can find this page. And please check language. And to study Japan, you can search school, but to study Japan, scholarship may be important. So please click here, and you can get many information. So about 500 information is available here. Many informations are available here. So search using the term study in Japan. And best option, and she also get this one, but the best option is a mixed scholarship. One of Japanese government scholarship. And this mixed scholarship has 2 types, university recommendation and embassy recommendation. And she got university recommendation type. And to get university recommendation type scholarship, connection with academic supervisor in Japan is essential. So making a connection is most important. And reaching out directly may not get a response. Many cases, now we got too many mail, so generally we may not have response. Okay. So it's better to have someone such as your current supervisor or Raju. They will introduce you. So please negotiate with your supervisor first, and your current supervisor in Japan. Please negotiate with your current supervisor in Japan. And in her case, she was introduced by Dr. Nadi, and she's familiar with me. So such connection is important. So I do not recommend reaching out directly. And this is important. To write mail, make sure to proofread your letter before sending it. So I got many mails, but English is not well written. This is a problem. So checking your letter is important to send mail. And in the case of university recommendations, factors such as established MOU or MOA between university is important. And clear short-term study experience in Japan is also important. And pre-existing collaborative research. And in our case, we have pre-existing collaborative research and MOU in our case. But if you have these are considered, these may be advantageous. Okay? And now we are attempting to contrast MOA between my university and Shiribishi Educational Society. So maybe you may get this chance. Okay, and after getting this chance, you may have some opportunity to study in Japan for short time period, such as 1 year. So because we have such program. Okay. So maybe we will establish this one soon. So wait for a while. Not yet, but we will get this one soon. So wait for a while. And for embassy recommendation, exams are important. And exams are a language examination, both English and Japanese. So to get embassy recommendation, a study of Japanese is also important. And there are some private scholarships. There are many private scholarships. So please check this page and you can find many. And I'm currently visiting here with researching fund from the Heiwa Nakajima Foundation. But this foundation provides some research fund, but main purpose of this foundation is particularly dedicated to supporting international students in Japan. So Nakajima-san established this foundation, but he was an international student from Korea and succeeded in Japan, and they made this foundation. So he contributed to support international students in Japan now. And other method is here. In Japan, international students are allowed to work part-time for up to 28 hours per week. So generally, we work 40 hours, but a student can work 28 hours. And many students use this system as a means to support their studies while studying in Japan. So this is one way. Work in Japan. Work and study in Japan is another way. And you can combine part-time job with Japan Student Service Organization Scholarship. And maybe it's money may be small, about 50,000 yen, so 100 1,000 rupee. So money may be small, but by combining this scholarship and part-time work, you may have some chance to study and work in Japan. And I want to introduce one example of employment in Japan. And she's Matthew-san. She's a member of my family, and now she introduced her. And now she got a position, and she will get a PhD in next March. And she got a position as a tenured researcher. Tenured is important. Tenured researcher at the National Institute of Advanced Industrial Science and Technology called IST. And IST and RIKEN are one of Japanese leading research institutions. So she got such position. So this one is one example. Okay, so one way is to go back to India, but you may get some jobs, some research positions in Japan, and this one is a typical example. Okay, so you have many chances to visit and study in Japan. So if you are interested in studying Japan, please contact me. Okay, okay, thank you so much.
Any queries? Oh, many. Mic akkada. Please maintain silence. Within 2 minutes, we'll wind up.
Do you have any queries?
Someone raised hand. Okay.
Good afternoon, sir. So, as pharmacists, what are our opportunities in Japan?
What?
As pharmacists,
Pharmacy student. I belong to agricultural faculty, but one of my interested in is medicinal plants. Okay, so one way is collaborated with me, and now I'm performing with collaborative research here about medicinal plants. So one way is to select me, but there are some pharmaceutical schools, so selecting them is one way. Okay.
So good afternoon. Do we need any entrance exam to enter into this college?
Sorry, please repeat.
any entrance examination required to get into it?
Do we need any exams
entrance
like
examination.
IELTS and all?
Please explain. She has examinations. But the problem is getting scholarship is important, and because such a person who get scholarship is high level, so don't worry about examination. Okay, but when you use last method work in Japan, examination was performed very strictly. So do your best. Okay.
In my case, I was selected as a university recommendation mixed scholar. So I only took IELTS. So I didn't want to get any other scholarship, any other exams. But when I extended into as a research student into master's student within the university, we need to take entrance exams. So in that case, we are foreign students. We don't need to like do written exams like that. We have to do only 3 minutes or 5 minutes presentation about our studies. But for the now I am extending my master to PhD, doctor course. So in that case, we have to do around 20 minutes presentation about our studies and future plans.
Okay. So generally, if you get a scholarship, it's not a problem. But if you want to work and study, exam is required and its level may be high. Okay. Any question?
Any more questions?
Okay. Thank you so much.
Thank you so much, Sensei, for throwing light on future education in Japan for my students. You may not be directly related to any research institutions related to pharmacy, but you can, this is all collaborative study. We are already collaborated with Sir in this Heiwa Nakajima project, as he has already mentioned. We are already working with him. So you may get into your higher education using this university education recommendation. There are two ways of getting into that university, right? So one way, if you are getting into an MOU with Sir, with Saga University, you can go to Japan for higher education using this university recommendation way. That process you can take up and you can get into higher education. And you can also, it's not that you are only restricting this way, you can link, like I said, he is working on medicinal plants. And I'll be working on the secondary metabolites. You all already studied about secondary metabolites and all active constituent. To improve the active constituent levels at an agricultural level, he'll be studying. So he'll be supporting us in that way. So you can relate your work with him and then work. And you might have seen that example of the PhD student. She got into the top research institution job. After completion of PhD under Sir, she got placement in higher, I mean, in Japan, it is the highest research institute. And another example is Mrs. Eranga. So these examples are enough for you to prove that if you are studying in Japan, you will settle well and you can improve your knowledge also. Thank you, Sensei, and thank you, Mrs. Eranga Pavani Vidarana, for helping my students to get knowledge on how their future would be if they are opting for Japan. Thank you. Third years can have lunch and come back by 1:30 for CRT program in the auditorium. That you can talk to Ram sir and make some arrangements, okay? Thank you.